organized by ISF College of Pharmacy, Moga, and sponsored by IBRO-APRC. Today, the task of introducing speaker is a great privilege. So, introducing our first speaker of the day, Professor Anil Kumar. So, Dr. Anil Kumar has joined UIPS in Punjab University as a lecturer in pharmacology in 2001 after serving to Dabur Research Foundation, Ghaziabad, and to New Delhi. Dr. Kumar has completed his PhD during this time along with teaching and aroused the level of professor of pharmacology. Dr. Kumar is currently youngest professor of Punjab University. Dr. Kumar, he obtained his postgraduate degree in M.Pharm and PhD degree in the field of pharmaceutical sciences with specialization in pharmacology at the Punjab University. Dr. Anil Kumar has started his career in the areas of neurosciences, special focus on neurological problems, particularly movement disorders such as Alzheimer's diseases, Huntington disease, Parkinson's, stress and related problems such as memory, stroke, sleep, and diabetes complications like diabetic neuropathy and nephropathy. Dr. Kumar's work has been cited extensively in national and international journals of repute, with more than 180 research papers, having H-index more than 42, citation more than 6,000. Dr. Kumar supervised several postgraduate, more than 40, and doctoral, more than 15 students. Currently, 6 PhD students are working under his kind supervision. 3 postgraduate students are also working in his laboratory. Dr. Kumar has received several national and international awards, such as ICMR Award for Biomedical Scientist, Chandrakanta Dandiya Prize for Best Research Publication in Pharmacology, Rashtra Ratan Award, Bharat Shiksha Ratan Award, Young Pharmacy Teacher of the Year Award by Association of Pharmaceutical Teachers of India, Dr. Ol Rifelson's Young Investigator Award from USA, LA Lilly Young Investigator Fellow Award from USA, AICTE Career Award for Young Teachers by AICTE, New Delhi, IBRO Travel Awards from IBRO, IBRO VLTP Award, Research Paper Award from Journal of Cell Tissue Research, DST Travel Award, ICAD Travel Award, ICTE DST Travel Award, more than 15 times. Boycast Fellowship of DST, INSA Visiting Fellowship, Riken Brain Science Fellowship from Japan, ISN, APSN, IBRO Fellowship for attending Advanced School of Neurosciences. Dr. Kumar extensively traveled abroad for various scientific activities like in Japan, Italy, USA, Australia, Malaysia, Thailand, and many more. So we welcome you, sir, for your valuable talk.
Thank you, Dr. Arti.
Yeah. Thank you very much, sir.
Thank you, Dr. Arti, for introducing me. So let me come directly to my talk. I'm audible, Arti?
Sir,
yes, sir.
audible? You are audible, sir.
Okay.
You can present now.
Okay. Arti, my slides are visible?
Yes, sir. They are visible.
Okay.
You can start.
So let me, first of all, I'd like to thank the organizer for inviting me and making a request to discuss the ethical issues. Although I was interested to discuss the neurological, but based on the request, I am just discussing these issues. Although I've discussed these issues several times and several platforms, but I think this is the issues which is need to know about the ethical aspects, whenever we do the experimental pharmacology, So that's what I brought these slides to you to know about what are the different ethical issues when we discuss or when we perform any experimental studies, as well as why these days the experimental studies are being discouraged and people are focusing more on alternative approach. So I brought these presentations to you about the different ethical issues, some of the guidelines which are generally nowadays focused on in India regarding CPCSCA, and some of the alternatives, right? So this presentation is talked about these 3 different aspects. So now let me start my story of that. You're all aware of that in pharmacology, we generally use the animal experimentations. We perform animal experimentation, and it's one of the important steps when of the drug development process. That it is not only the preclinical studies, but preclinical studies are basically that include so many things are there. You can see it. Nowadays people, they are doing the preclinical studies, toxicity, acute toxicity, subacute toxicity, chronic toxicity, and that includes not only the in vitro, in vivo studies, but other many more studies are being included. And when we talk about the biological experimentations, so ethics comes into the picture. And these days, when the different ethical bodies, they generally discourage the use of animal experimentations, and they are focusing more on the alternatives. Now, that is what I said, that it is, okay, before discussing this, that. So when we do preclinical studies, the animal model is one of the most, important steps in all the experimental pharmacology. And that whenever we do anything, the animals is basic tools to prove our hypothesis preclinically. And that is what you can see there. So many things are there, either to target to hit to lead to candidate, IND investigation, new drugs. So many things are there when we do the preclinical work. Out of this, all we need to prove it with the help of the animal models. So animals are the basic tools that is generally used in the preclinical experimentations. Now, that why these days the animal experimentation is generally considered as a difficult issue. That although we know that the animal experimentations are widely used to screen the new medicines or new molecules, new lead molecules. And when we talk about the experimentation on the animals, we generally induce the disease condition in the animals. That include the lot of pains are there. And sometimes we say it is morally incorrect to induce the disease condition in the animal, which is not clinically relevant, or they are not truly translating the clinical situation in the animals, because every animal models has certain limitations, right? Every animal model has certain unique features. And based on this, we generally use the animals for our hypothesis or to screen the new molecules. So these days, it is a big concern about the ethical issues. What is that ethical issues is, when we use animals, it is very important to justify the experiment we are conducting on the animals, right? Either whatever the animal we are using or choosing for our experimental hypothesis, it is very important that our experiment must have strong rationale to prove our hypothesis. So that is what it is very important to justify the experimental procedure ethically. And that is what we say it is very, very important challenge for the scientists to prove that the experiment that we are conducting or that we are proposing on the animals is rationally justified. Now, this slide talking about the why, and if you see globally, you will find a lot of animals are being currently used also for the preclinical research. If you see, one of the data says about 70 to 100 million vertebrates per year are used in the either for research or testing of drugs or vaccines. And out of these, 30% of the animals are used for the purpose of fundamental research and diagnostics. And mice and rats are frequently used for among these animals, And even in many European countries, you will find nowadays European countries also making a lot of rules and regulation to control the experimentation on animals by making different laws. And also they are making the laws or rules, regulations to ensure the ethics that is being followed in the experimentation. And that is what we are more concerned about the animal experimentation these days, And now why the people are more concerned about the animal experimentation. You will find that whenever we conduct the experimentation, we generally keep the animals in the captivity. They are unable to escape. We generally induce the pain and distress. Sometimes the animal die during the experimentation. Sometimes we poison the animal, deprive of food, and psychological stress. We generally induce sometimes infection with the disease. So these are all conditions we generally induce in the animal to prove our hypothesis. And if you see all these lists for a layman, it is just a list of torture that we generally do on the animals without any scientific rationale. So that is what nowadays very, very much concern is there. We need to ensure the welfare in the experimentations. So when we talk about the animal welfare, it implies that whenever we use the animal for the experimentation, it must be free from hunger and thirst. It must be free from discomfort. It must be free from pain, injury, and also the animal should be allowed to express their behavior. It should be free from fear and distress. These are all comes under the welfare. So it is very, very important for the scientist who is conducting experiment that must ensure that the animal welfare is taken into consideration while performing experimentation on animals. Now there are so many people are there who are in favor of experimentation, and some group, for example, in India, there is a PETA organization is there who is against the experimentation on the animal. Even if you see in India, there is a UGC, University Grant Commission is there, who is also discouraging the use of animal experimentation for demonstration purpose. So there are two views I have given this slide. That is the one group which is in favor of the animal experimentation that says that it can be, allowed if the suffering is minimized. It means the pain and suffering can be minimized in the experimentation, then it can be allowed. Second one is the human benefits are gained which could not be obtained by using other models. Because we are just generally making the drugs, we are discovering the drug, we are screening the drugs for the benefit of the human beings. So the people who are in the favor of experimentation, they says you can use the experimentation or you can do the animal experimentation, but provided it must be justified, it must have minimized pain, and it must be benefit to the human beings, right? The another group which is against the animal experimentations, they said we should not unnecessarily cause suffering to the animal without any scientific backup. If the experimentation is not having scientific rationale or the basis, it is waste to sacrifice the life of the animals. So if the benefits to the human beings are not proven, then there is no need to conduct the experiment just for demonstration. So that is what in India also the demonstration for the animal experimentation for demonstration purpose is not discouraged or totally stopped. Now, if you see the comparison versus favor versus against, you will find, so what is the situation when we can use the animal experimentation? So animal experimentation can be allowed. We need to see the benefit versus risk. If the benefit is more, then the animal experimentation should be allowed. And if we are allowing the animal experimentation, we should have a strong justification for the use of the animal. The experimental procedure we are using, it must be ethically sound. So these are the three key points which need to be taken into consideration while getting approval for the animal experimentation. Even if you see the other legislative regulation on animal protection, you will find the first rules, even in India also, the Prevention to Cruelty Act is there in 1960. But even in the UK also, the first law, the Cruelty to Animal Act, was adopted in UK in 1876. Even then, later on, the two European document which is there in the net also, that is talking about the welfare of the animal. And now these bodies or these acts and rule regulations are very, very stringent when we conduct the animal experimentation on animals. Now, in India, we generally follow the CPCSA guidelines. That the full form of the CPCA is Committee for the Purpose of Control, Supervision on Experiment on Animals. So this is one of the regulatory bodies in India, which basically constituted under the Act of Parliament in 1991. And the main CPCSA is basically come out from the Section 15 of the Prevention to Cruelty Act 1960. So this act is basically this CPCSA is basically mentioned in the Section 15 of this act. Now, what these act talk about? This act, under this act, there is a rules are given. So rules for the animal experimentation is given in the CPCSA, which is notified in 1998, and then it was amended in 2001 and 2006 to regulate the animal experimentation on animals. So in India, currently, the all animal experimentation, starting from small animal to the big animals, is taken care by the rule regulation of the CPCSA, whether it is industry or whether it is an academic institution, whether it is a commercial institution. So all the experimentation on biologicals are controlled by CPCSA. So it is very important to understand the rule regulations and norms set up by this CPCSCA. Now, this is a slide which is talking about the Prevention to Cruelty Act. You can see under the chapter 44, the section 15 is there, that is Committee for Control and Supervision of Experiments on Animals. So section 15 talk about the CPCSCA. So you can see this is not new, but this section 15 is just come out from the Prevention to Cruelty Act. That is what is there. So now this CPCSCs talk about a lot of regulations. Now recently in 2018, the CPCA has further revised. So the recent compendium that is appeared in the website is of the CPCSA is 2018, which talk about all the compilation of the guidelines of the CPCSA related to the animal experimentation. So in the coming slides, I'm just talking about the few points, key points of the guidelines that is very, very important for the scientists to know who was involved in animal experimentations or who are planning to conduct the experimentation on animals. So what is CPCSA? What are the major objectives and functions of the CPCSA? So the main objective of the this body is to ensure that the animals are being used and they should not be cause any unnecessary pain and suffering during the experiment or after the completion of the experiment. That is what the main objective of these guidelines. So you need to ensure as a scientist that whenever you conduct the experiments on the animals, these experimentation should not cause unnecessary pain. If the pain is involved, one has to use the painkillers. That is what the ethics says, right? Apart from this, the CPC has several other functions which is laid down in the section 17 of Prevention to Cruelty Act, and that is talk about the registration of the institution. So CPCSA currently not only regulating the experimentations, but they also controlling the registration of the institutions which are conducting experiments. They are also ensuring that experiments are conducted by the qualified person. They also monitor and inspect the experimentations with the help of their own nominees. They provide the permissions to register the institutions if they fulfill the requirement or the criteria to function as an animal house. Apart from this, they regulate the experiments on the animals. They monitor the transfer or acquisition of the animals for experimentation either from the outside the country or inside the country from one institution to others. Apart from this, CPC also nominate their members or trained members to perform their work in the meetings of the Institutional Animal Ethics Committee, where they generally regulate or they ensure the ethics has to be ensured in all the experimentation and the issue of the or approval of the animals. So they also conduct the training programs of different peoples to create or to increase the manpower of the bodies. So in the form of nominees, they give the assignment to different institutions who are conducting the experiments on animals. Apart from this, CPC also conduct the workshops and conferences to ensure to promote the animal ethics. So these are some of these the functions of the CPCSAs are currently laid down in the website also. Apart from this, they also provide the legal or penal provisions if any establishment violate these norms. Right. Now comes to the basic ethical principle in scientific experiments. Why it is important to discuss these ethical principles? Because most of the young scientists or young researchers are not aware of. Although we are in India, the animals experimentations are banned for demonstration, but there is no ban for experimentation which is required on animals for research purpose, right? But these are the criteria one has to know that on what grounds you can take the sanction of the animals for the experimentations. What is that experiment grounds? It means if you are asking the animals or for experimentations for the purpose of advancement of new discovery or knowledge, which is not existing in the literature. That is one criteria. Number two, the knowledge which is expected to be useful for the saving of the humankind. That is also the criteria. For prolonging the human life or alleviating the suffering. For example, you are making the new drugs that will delay the survival of the human beings during, for example, in the case of cancer. So that drugs, if you are testing on the animals, that kind of experimentation can be the ground to justify that the experimentation is rationally required to conduct the experiment on animals. Then, if for the significant gains in well-being of the people of the country, for combating any disease, whether of the human beings or animals or plant. So these are some of the criterias are there where the animal experimentation can be justified. So this justification has to be there when you ask or seek the animal for their experimentation. Second one is, if you are asking the animals, it is better to choose the animals which are lowest in phylogenetics. What it means? It means the animals which are lowest in phylogenetics have a less degree of sentience. What is sentience? Means the feeling or perception. The animals as small as the animal is there, the feeling of expression is less as compared to the larger animal. So nowadays, that is what the people, they are using zebrafish, they are using Drosophila, lowest in phylogenetic as compared to the rat, mice, monkey, guinea pig, all this. So it is very important, if you want to choose it, it is better to choose the animals lowest in phylogenetics so that you have a less degree of sentience, and that is what is justified. Right? So these are the animal experimentation which can be subject to the regulation. For example, you can start from the invertebrate to the vertebrates, and these are there. The third principle, which is very, very important, and it is globally recognized, is the 5R concept, which was earlier, it was 3R concept. Now it is reversed to this 3R to 5R concept. It is growing, So what is this 5R concept is there? That is called as the replacement, reduction, refinement, rehabilitation, and reuse of the animals are called as the 5R concept. So let's discuss first one by briefly. What is replacement, reduction, refinement, rehabilitation, and reuse of the animals? Now, if you know the replacement, it means if you need to use the animal, it is very, very important that you must replace the conventional or older models with the new non-animal models as far as possible to justify or to fulfill the scientific aim and objective of your studies, if it is possible, right? Or either you can replace the larger animal with the small animals, or you can replace, with a cell or tissue of the animals. For example, you can take the absolute replacement, larger animal with the small animal, that is called as absolute replacement, or you can take the relative replacement, where you can use the cell of the animals or tissue of the animals, that is called as relative replacement, instead of wasting the entire animal life. So that is what is the replacement people they're using, and they're using this concept so that we can reduce the number of animals suffering in the experimentation, right? There are so many examples are there where the people they are following and adopting the replacement. For example, people they are using the in vitro techniques. It is also one of the kind of the replacement. Replacement means a higher animal with the lower animals. We can use the computer-based model. So these are the different models which can be used as a replacement to justify the aims and objectives or justifying the experimentations. Now you can see the advantage of replacement. You can see the replacement is generally based on the pre-existing knowledge for teaching. For example, we have developed so many software, right? So software is basically based on the pre-existing knowledge, and based on this, softwares are generally used to demonstrate different effect of the drugs, maybe agonist, antagonist, maybe competitive, non-competitive, maybe DRC, maybe CRC. There are so many concepts can be explained with the help of computer-based models as a replacement, instead of using the life of the animal or sacrificing the animal just to demonstrate. And that is what is one of the replacement. Although every replacement has certain limitations are also there. For example, the new models are based on the pre-existing information, right? But the living system is very complex system. So it is some disadvantage or limitation is there, but it does not mean that replacement cannot be adopted, right? So although absolute replacement is not always an option, right? That is what is there. So limitation is there, but replacement can be adopted. Similarly, one of the example of alternatives or replacement is, which can you see that long years back, when the woman wanted to know whether she was pregnant or not, we usually used a rabbit test. But nowadays, you all aware there is a pregnant kits are there, which can be used to know whether the female is pregnant or not, whether she conceived or not. So this test is generally replace the earlier rabbit test, which were used to kill the rabbit, So this is one of the very, very right replacement that will giving you more edge to the scientific community and giving the more validity of the results as compared to killing the animals just to know that whether female is pregnant or not, right? Similarly, the second concept R is the reduction. If you cannot replace the animals, small or large, can you reduce the number of animals in the experimentation? And that is what the Institutional Ethics Committee always ensure. Always try to reduce the number of animals in the experimentation as far as possible. And this can be done by doing the pilot studies. This can be done by using the preliminary studies. This can be done by using the half of the group so that you can have some more confidence before actually being used the all animals in one group to fulfill the objectives. So that is what is there as an important concept of second R, which the scientists should know it before it is being used or before it is they start using the animals, all animals in one group, right? Third one, so there are so many strategies are generally used to reduce the number of animals. For example, we can do the pilot studies, we can do retrospective analysis, we can do ethical review, we can reuse the animals, we can do the harmonization of the guidelines are there. So many strategies people they are using to reduce the number of animal so that we can reduce the unnecessary pain in the animal or animal experimentations. Third one is the refinement. Now, this is very, very important nowadays. People, they are saying, or researchers, they are saying, it is very, very important to refine the experimentation. You must have read a lot of research papers where it is written that it is a modified method, right? So when we say it is a modified method, it means you are refining the experiment of the previous established experiment. By refining, you are causing less pain or suffering to the animals. For example, the earlier you must be aware there is an open heart surgery was there, but now it is a bypass surgery there. So bypass surgery is also an example of the refinement, So that will cause less suffering to the human beings. Same refinement concept is nowadays using in the animal experimentation as a third R concept, and we can do it by refinement. For example, we can use the minor sophisticated techniques so that it causes less pain to the and trauma to the animal during the animal experimentation. Now, the fourth R and fifth R concept, which is also growing nowadays, that is called as rehabilitation. Fourth R is the rehabilitation of the animals. Earlier, the rehabilitation concept was used for the larger animals, but nowadays it is said that it is a responsibility of the PI to rehabilitate the animal after the experimentation is over. It means after completion of the experiment, it is very important. It is a responsibility of the PI to take care of the animal till the animal die by their own natural death. And this cost has to be included in the project proposal when they submit proposal for their financial sanction or financial subsidy, right? And fifth R is the reuse of the animal. So based on the drug that is being used in the experimentation or has been used in the experimentation, the animal can be reused after washout period, and that is called as fifth R concept. So if you are using the animals, these 5R concept has to be applied in the experimentations, and that is very, very important as a basic principle of the ethics in the experimentation. And third principle is proper use of animals in experimentation. Now, this is very normal generalized statement, but it has a very, very strong implication. What it means? It means if we are using the animals in the experimentation, you need to take care of that the animals should not cause unnecessary pain and suffering. If the animal is having a pain and suffering, you will never get reproducible results. Result can never be reproduced. And you must be aware that a lot of drugs, they fail during the preclinical studies because they are not producing reproducible result. Why? Because when we do use the animals, they are either not in the proper care, and that is what the results, they are not reproducible when another researcher, they carried out the same studies with the same conditions. So it is very important that when you use the animal, it must be carried out under the same analgesics or anesthesia or sedation or painkiller as far as the conditions required in order to ensure the ethics. Therefore, that whenever we conduct the experiments on the animal, sometimes we need to take care of the anesthesia. Sometimes we need to sacrifice the animal because we want to take the liver, heart, brain, or different tissue of the animal. So it is important to sacrifice the animal. So CPCSE says you need to use the anesthetics which is approved by CPCSE, right? So the guideline says these are the anesthetics that can be used during the experimentation to induce anesthesia or euthanasia in the animals. Now you can see these are the guidelines which talk about you can use these 4 anesthetics: ketamine, pentobarbital sodium, thiopentol, and these are dose has been given in the compendium 2018. So which is now they have mentioned the dose. Earlier there was no mention of the dose, but now they said you need to use these only 4 anesthetics whenever you perform any anesthetic procedure in the animal experimentation. If you write down something else as an anesthetics, your protocol cannot be approved by CPCSE as per the norms. That is what is important. So you need to know these anesthetics in mind whenever you perform any experimentation involving anesthesia. Now, most of the times you must be aware of that when the ketamine is generally used in combination with the xylazine, you're all aware. And you must know that these ketamine should not be used alone. That is what it is given recently in the guidelines, that ketamine should not be used alone because it causes a problem to the animals. So it has to be used along with xylazine. And you must be aware in most of the experimental procedures, even 99% of the experimental procedure where the anesthetics are being used, it is generally used along with the xylazine. And why? Because in small animals, if you use only the ketamine, it may cause lameness in the animals. So that is what ketamine is always used along with the xylazine, right? That is what you need to know. These all, Apart from this, when we talk about the euthanasia, euthanasia means sacrification of the animals. So there are different methods have been given by these bodies that it can be allowed. So NR recommends, that is called as not recommended. Talk about the allowed. So you can see in the case of mouse, you can do decapitation or cervical dislocation. Now this is a new point which is added recently is in the case of rat, you can see the star here. If the rat is having body weight greater than 200 grams, then cervical dislocation is not allowed. Be careful. If the rat weight is greater than 200 grams, then cervical dislocation is not allowed as per CPCSE norms. It was not there earlier, but now it is mentioned that if the animal weight is greater than 200 grams, cervical dislocation is not allowed, right? That is what one has to understand. If the body weight is less than 200, you can do it. Otherwise, you need to do decapitation or other method which can be allowed by CPC. For example, you can give the barbiturate in high dose, chlorohydrate, ketamine, sodium pentothal you can use, or you can use the carbon monoxide, carbon dioxide. You can use it as a anesthetics, for euthanasia or sacrificing the animals. But you cannot do cervical dislocation in the animals having greater than 200 body weight. Now, these are the methods which are not allowed. Most of the time, sometimes the people, the researchers, they use because they are mentioned in the research papers of older years research paper. If you are following, they are generally using that stunning hair, inhalation of gases hair. These methods are totally now banned. It is not acceptable as ethics point of view, right? So methods not acceptable for euthanasia is the decompression, stunning, nitrogen flushing, organ flushing. These are all methods are totally not acceptable, or ethically they are not sound. So that's what they are not allowed as per CPCSE norms. Now the another basic principle is moral responsibility. So it is also mentioned in as ethical point of view. If you are using the animals for your experimentation, it is your responsibility to take care of the animals in all ways, even after the experimentation. That is what is the moral responsibility of the researcher, to take care of the animals till they die by their own natural death. Next, The another principle that is called as a living condition. Now, why we are discussing this is a basic principle, because if the living condition of the animal is not proper, animal will not behave normally, and you cannot ensure the results that is produced from the experimentation will be reproduced, because if the living condition of the animal is not proper, the animal will not behave normally, right? So that is what. Then physical facilities, right? So physical facility is also the another facilities of the central the animal house facility, which is given in the animal house. This is very generalized, but every point has their own importance, why, although it is not that much they need to discuss, but you must know that these are the point which is very important to ensure that your animal house facility must have, because they have their own importance, and that will be important for making the health of the animals sound, right? For example, building material, corridor is there, utility is there, animal door rooms are there, floors are there, because these are the points which are very, very important, and they have important role in the welfare of the animal, right? Now, these are the important points in the case of animal house facility when we talk about the temperature and humidity. Now, why it is important to discuss the temperature and humidity? Because you need to know if the temperature and humidity is not controlled in the animal house or the facility where the animal are housed, you can think of the animal hemodynamic parameters will change. And when the points or the parameters change, your results will definitely change, and you will never get reproducibility result when you repeat the experiment under the different set of conditions. Next is the ventilation. Ventilation is also important for the better health of the animals. Power and lighting is also important, because the animal need the dim light, Now you can think of if you can you sleep in the on the night when the light is on, you will never get sleep, because that is the environment is required for the animal to feel comfortable as well. So for power lighting is there. No, you must be aware of that the animal require 300 lux light. They do not require the high light, as we require. So animal require 300 lux light. They require 24 hours power backup supply, so that the temperature and humidity conditions can be maintained. Noise control is also important for the psychological health of the animal, because always we recommend that animal house has to be at the corner place, so that there should not be unnecessary noise, because if the noise is there in the animals, the animal can never behave normal, So these points have their own importance, and as a scientist you should ensure that the animal should behave normally, and it can be done only if these conditions are fulfilled, Similarly, the housing or caging system or animal husbandry. So whenever you keep the animals, it has to be in the social group, It has to be provide a comfortable environment. Food and water, we generally provide the hygienic food. We provide the RO water to the animal. We provide the ventilation in the animal house, right? We provide the temperature, humidity. We provide the clean cages. Everything is there. So these are the environment has to be created, or this facility has to be provided to the animal house or to the animal, so that the animal behave normally, right? So because the animal is a social animal need to live in a social in a group, So when the animal lives in a group, they communicate each other, right? So this CPC says that the animal should be, if less than 10 grams of weight, it must have this floor area. If the animal is up to 15 grams, it requires this much floor area. So these are the guidelines have been given by CPC, so that any scientists or researchers or who are taking care of the animal house must ensure that these animal must provide the proper floor or space, so that the animal behave normally, right? Similarly, those animals which are bigger in size, they need to provide the shelter or outdoor facilities, right? Similarly, the social environment, as I said, the animal required to communicate each other, so they always animal required to live in a group, so they always keep in group. If you keep the one animal in one cage, so you will find the animal will become a social isolation stress, right? So the animal need to provide the the enrichment environment in the cage so that the animal feel psychological or physical health. So these are the important point which is given by the CPCC guidelines to ensure the proper health of the animals. Similarly, the activities has to be there in the animal house. For example, in the cage, we cannot open the animal, so we provide the enrichment to the cages. For example, the paper sheds are there, some toys are there, pipes can be kept in the cages, so that the animal feel normal. Similarly, the food, we generally provide the palatable, non-contaminated, nutritionally sound feed has to be provided to the animal, right? Similarly, the bedding material is there. Nowadays, corn cobs are used. The other bedding materials are there. So the animal has to be, the guideline says you need to change the cage twice weekly. So that the animal must be provided the dry bedding material so that the animal can perform, its vital functions. Similarly, the water has to be given twice daily. At least RO water has to be given. Water report has to be maintained, so that you can say that the animal is being given the RO water as per the requirement or norms given by the CPCSA. Sanitation, cleanness is there. Waste disposal is there. Weekend holidays care is there. So these are all points which is mentioned or covered in the CPCSA guidelines. That is important. One has to know about these points as a researcher, because these points also impact the health and behavior of the animal. And if they are not ensured, you cannot say that your animals is normal in behavior. So that is what is important to know. Now, this is another important point regarding the animal house facility, that is called as record keeping. You all should aware that these record has to be in place in animal house, whether it is a Form C, Form, the sick record, training record, water analysis report, health record of the animal house, animal record, SOPs. These are all record has to be there as per the norms of the CPC. So the CPC normally generally ensure all these points has to be in place whenever they do inspection of the animal house. That is what is there. Similarly, the guidelines talk about the transportation of the animals from one institution to other. So they said whenever the animal has to be transported, the animal transport vehicle has to be shock free, right? It must have a sufficient space so that, and food and water has to be there in the cages. When you transport animal from one institution to another institution, there should not be unnecessary stress. There should not be unnecessary jerks, right? The transport vehicle must have the facility where temperature and humidity has to be maintained and the food has to be provided. That is what is the minimum requirement when you transport the animal from one institution to another institution. Even the CPCSA also laid down, when you transport the animal from one institution either by road, rail or air, you need to provide this much space to the animal. For example, mice, if they have a 25 mice in one cage, it must be of the metal cardboard transport box has to be there. That must be have a space of 20 to 25 centimeter square is there. Height of the box must be 12 centimeter. These are the guidelines has been given by the CPCSA when we transport the animal from one institution to another. Now, based on these normal guidelines, principles of the animals and justification of the ethics, it is important now people are or researchers are now moving or shifting towards the alternatives. So this last portion of my talk is basically focus on different alternatives these scientists are using. So the young researchers should know there are a lot of alternatives are there. So whatever these alternatives I'm giving, it is not a complete list. Right now, there are more than 3,000 alternatives are available, and that can be used in the research to fulfill the aim and objectives. But researchers should know that these alternatives are exist and they can use these alternatives for in their research, in their experiment, and that is the totally replacement of the animals. And why these alternatives are being now encouraged, because they are more reliable than the animal test. They are less, the environment, they are more environmental friendly. They are truly free product. They use the human toxicity in toxicity testing. They are more accurate than the animal models, And because of the advantages are more based on the alternatives, That is what nowadays alternatives are being encouraged in all places, that is what. So you should know, even if you see that whatever the data we are collecting preclinically on the animals, they need to be generally submitted to the regulatory bodies to get the product approval. Even that is what now the different regulatory bodies of US, Japan, European Union, they're also encouraging, they are accepting the alternatives to the animal testing. Now you can see in the ICH guidelines, OECD guidelines, and other guidelines, the alternatives are now widely accepted. So the researchers should know that these alternatives are now reliable and acceptable among the regulatory bodies. So one has to know and use these alternatives as a reliable model, right? So alternatives are generally used either in the research, testing, and academics. These alternatives are generally used. These are the few areas where the alternatives are used. Now, when we talk about the research and testing, we generally use the epidemiological studies as an alternative. What is epidemiological studies? We generally collect the information in the population, and that will help to justify or to create the hypothesis of any research experiment. So before actually conducting the experiment, it is very important to collect the information. So epidemiological studies are another alternatives, which is a tool that can be used to generate the hypothesis before actually conduct of the experimentation. For example, if I say that smoking causes cancer, for example, right, it is well known established fact. No need to create the cancer in the animal by inducing by giving the smoking to the animals, where it is well known fact is there, right? So there is no need to create the same kind of things which is already there in the scientific domain, So these are the things which is information that can be collected from the population-based studies, and that is generally used to generate hypotheses and can conduct the experiment. You all aware of the importance of cell and tissue culture techniques. These days, these are widely used as a technique, and there's no need to discuss all this because cell and tissue cultures are widely used as alternatives in testing of different types of studies. For example, mutagenic potential is there for the product ingredients. They are used for the anti-cancer, anti-AIDS, neurological problems. Currently, cell and tissue culture techniques are being used as an in vitro technique in almost all the area of research. All area. Name anywhere where these are not being used. So almost see, everybody is using the cell and tissue culture. So one has to be equipped with the cell and tissue culture technique as an alternative. Although the cell and tissue culture have their own limitation. Why it means? Because the maintenance of these cells also require the animal derived serum. Although they require, but you can say that the animal sacrification can be minimized to a greater extent if we are using cell and tissue culture technique as a model or as an alternative to fulfill the hypothesis. There is another, which is a slide which I have shown several times, that is a epi-skin model. Why I'm discussing this epi-skin model? Because this is a model which is widely accepted among regulatory guidelines. And this is a epi-skin, which is a in vitro reconstructed human epidermis, which is generally used as for different testing of the skin-related testing. For example, corrosive, irritative studies are there where these are used. Now, the success of this epi-skin model can be, see. This model is currently used to evaluate the safety assay, efficacy assay, in different types of the guidelines it is mentioned. Now, that is what the success of this model. That is why it is being now used in different guidelines as a part to ensure the safety. So one has to know that this model is there so that they can use it whenever they require, instead of using the traditional animal skin. Now, this is another corrosivity test, is an in vitro test system is there that is generally used to determine the chemical corrosivity. Now, this is what is there. This is basically a replacement of the rabbit eye test, which was earlier take several weeks, and it was the expensive test which generally replaced, So it is a corrosivity test is basically proprietary biomembrane chemical detection system, which generally gives you the colored when any corrosive substance comes into picture. So this advantage is it saves time and money over the traditional methods. There is another one is a chitosan film is there, which is used to different types of, you can say drug delivery system. So this film is generally a animal, is a substitute of the animal, a human epidermal sheet, and it is generally used for as a local delivery system to check it whether the drug is being delivered properly or not. Although so many factors are there which generally affect the delivery or the system, but this system is there which can be used to study as compared to the traditional animal skin. Similarly, there is you all aware of in Parkinson, we generally induce the disease in the animals either by NPTP model or 6-hydroxydopamine model. So many models are there which generally induce the Parkinson. But nowadays, the human dopaminergic neurons are there, which is more reliable model, which give you more concrete information as compared to the model which is we induce the disease condition in the animals. Why? Because these human neuronal models, cell lines models, they generally give the expression of the tyrosine hydroxylase, which will be augmented to the human brain cells. So based on these human dopaminergic neurons, we can save this suffering or sacrification of so many animals. Now you can see that these dopaminergic neurons, they can save at least 1 lakh of the animals if this method is used to test the anti-Parkinsonian activity of the drug. Apart from this, there are so many cell lines are there which is generally used in different research program. All you must be doing different type of research, and different cell lines are there, right? That is what the people they are using. You all know the success of the stem cell. That is also there. Stem cell is widely used. It is because it is also used in clinics also these days in different neurological problems. The stem cell being used to regenerate. Although stem cells have their own limitation, but stem cells success is well proven. No need to discuss all this, but these alternative systems are there which one has to use it in their research as compared to the animals. These are some of the in vitro tests are there in the literature. I'm not going to go into the details, but these systems are there which can be used in your experimentation. Whenever you perform or plan an experimentation, always go and check whether any alternatives is available for your experimentation or not, right? That is what is the important. Nowadays, people, they are using zebrafish. You're all aware. The success of zebrafish is wide nowadays. People, they are using zebrafish like anything nowadays. And if in the coming years, you will see that zebrafish will totally replace the mice and rat very soon. Why? Because the zebrafish has been proven for different types of investigations. Cancer, heart, neurological, different types of disease conditions. Zebrafish is giving a very reliable results, And you can see why. What is the unique feature in zebrafish? Because it is a transparent body. You can see the organogenesis in the zebrafish easily. You can monitor. You can do the testing, toxicity testing in zebrafish. You can do the neurological estimation in zebrafish. You can see zebrafish can be used to evaluate the QT interval, which is not possible earlier. Earlier, we used to do the dog experiments, to assess or to study the QT interval, right? But now zebrafish is a very reliable model to study the QT intervals. And recently also in 2021, the CPCSE also laid down the guidelines for fish, zebrafish. Different guidelines are there. You must see and download the guidelines for fish, which is laid down in the by the CPCSE in 2021, Right. So I'm not going to coming to the details of the fish because this is separate, but they have given the guidelines how you can maintain, how you can preserve the fish, what kind of toxicity you can do, how you can maintain the ethics in the during the fish experimentation, how you can euthanize the fishes. These are all is mentioned in the CPCA guidelines. You must see and download at least these 60-page guidelines are there. That is what is important to know. Now, in the case of Drosophila, you know that these are also invertebrate and using as alternatives. Why? Because the lifespan is in 4 stages: embryo, larva, pupa, and adult. And you can think of the embryo is widely used. Why? Because you can see it, how these embryo, larva, pupa, how their development stage is there. Lifespan is short. And you can do all kind of genetic studies in the Drosophila, which is not possible or which is take a lot of time when we use rat and mice. So these Drosophila is currently being used in different types of genetical diseases, for example, Alzheimer, Parkinson, Huntington. These are all have a genetic link, So these are the sensitive model to study different complex disease conditions, and people they are using it. And another advantage is the cost of keeping this Drosophila is very less. Maintenance and propagation is very cost effective, and that is what you can maintain in a small lab. The Drosophila, you need only microscope and the Drosophila, right? Simple in the boxes, small box you can preserve or you can maintain it, C. elegans is another alternative which is used, Now you can see the C. elegans is a, although it is not widely used, but it is there in the as an alternative where embryogenesis occurs in 12 hours, adult is formed in 2.5 days. So the lifespan is short. You can do all kind of genetic studies. You can do neurological estimations with the help of C. elegans. The idea of discussing these in a short is to know that these alternatives are exist. People, they are using it because we are not aware of these models as an alternative. That is what we are reluctant to use it, Apart from these alternatives, different organisms, different microbiologists, they use these organisms for their different models, So these organisms are also there as a model because different organisms, they are involved in different pathways, So they can be used in different types of estimations, Similarly, there is a microorganism, for example, Saccharomyces cerevisiae is there, which is an important model, and it can be used to maintain the media, genetic system, DNA transformation system. These are all where it is being used, right? Apart from this, the patch clamp is another model. Although the cost of patch clamp is very, very high, but patch clamp is also an independent in vitro system where you can do single cell recording. And this is the model which is widely used, but it is not so easily accessible because of the cost, but it is there, Now, apart from this, there are different computer-based alternatives are there. Different chemistry people, they always use QSAR techniques are there. Sector activity relationships are there. And different pharmaceutical industries are using these days computer-based alternatives. For example, in silico methods. All researchers, they are using in silico methods. Why? Because they give the genesis of in silico method is they generally see the properties of the model. If the basic group is same, they may having the same properties, in the subsequent compounds, So they always see that the substance with a similar chemical structure or base or the functional group, they may have similar property. That is the basis of in silico method, So different in silico methods are being generally used to find out the prediction that these compounds may have XYZ property if they have same functional group or basic moiety. And these in silico methods are the preliminary studies to get the idea whether your compound may or may not have the activity, right? So there are different in silico models are there. Predictors is there, virtual docs lab is there, quick palm is there, pharma expert is there. In silico methods are there, Some of them they are paid, some of them they are the free models are there that can be used, Now this is another which is very highly productive technique that is called as high throughput screening, and it is a synergy of the chemistry, biology, engineering, bioinformatics, right? And the success, you can assume that number of drugs belongs to the category of AIDS, cancer are produced from this technique. You can see success is because you can screen 1 lakh compounds in a day, which is highly impractically impossible through the animal experimentation. And this high throughput success, you can see number of drugs are there in the market which are produced or which are obtained from this technique only. And that is what industry is currently rely on this technique very, very high use. Apart from this computer aided drug designing, nowadays you know that there are so many drugs which are in different isomers, they are more effective. So computer aided drug designing where you can design your drug based on their functional group, based on their receptors. So these are the another application software based technology people they are using. They are designing the drug based on the receptor structure, right? That is what is there. Now, apart from this, we can also use different clinical studies. For example, cancer tissues we can take, and that can be a starting material to do the research. For example, autopsy and postmortem studies are there, right? So people, they use different the surgical tissues and they do their research from there. What are gene expression, what are the receptors expression in different types of the brain tissue or the tissues of the body, right? So these are computerized patient database is there, post-marketing surveillance is there. These computer technologies generally collect a lot of information that can be the basis to decide or to design your own research. So these are not only that the only animals are the sole method to do research. You can do so many ways the research in different ways depending on whatever the things you have in your lab, right? So there are different non-invasive imaging techniques are there which can be used to fulfill the objective for your research. For example, computer tomography is there, MRI. Although these are all used also in the clinics also, but these techniques are also used, can be better interpretation of your experimentations. You all aware of microdosing, because when we do clinical research, we are all aware of the phase 0, 1, 2, 3. So microdosing generally give you the idea about the prediction of the toxicity when we use the 1/100th of the dose, pharmacological dose that is to be used on the human beings. And microdosing is one of the area where we give the predictions, what kind of toxicity may produce in the human being or during the phase 1 trial. So people, they are using the microdosing, right? There is another one which is recently I came across, that is a trauma man. You all aware traumatic brain injury is another situation where we do not know the exact drug. It is always treated based on symptoms. So nowadays there is a computerized trauma man is there, which is basically provided the prediction if the accident occur, what kind of surgery will be there, and if the impact is there, what kind of the surgery or what kind of the problems we may occur. And we generally use in the military training or training medical students, how to deal with the trauma when the patient suffer from different type of traumatic conditions. Apart from this, you can see these days people, they are using the artificial intelligence. Artificial intelligence is another program which is being used in drug discovery. Even drug development, the people, they are using the artificial intelligence, machine learning. These are the number of tools are people, they are using. This is all basically more reliable and it is alternative to the animals, simple, straightforward. So these are all about this, So in summary and conclusion, you can see the use of animal in experiment or research testing is not a right, but a privilege, right? So it is a responsibility of the every researcher to ensure that animal must be used ethically and the experiment is scientifically sound and being used rationally. That is what. And it is also same is ensured by the ethics committee all the time, right? Animals should not cause unnecessary pain. Pain must be justified. Pain must be cured with the help of painkillers or analgesic agents. If you are doing surgery, use the anesthetics, That is what is their ethics, right? So the animal should not be used unnecessarily. It has to be rationally justified, So every step has to be taken to reduce or follow the 5R concept, right? So this is what I'm saying every time. Try to use the alternatives. If alternatives are available, better to go for the alternative instead of using the animal. Animal has to be used in the last, right? So in the last slide, if you see, I'll show you this slide. It's very good slide. You can see the trend has totally changed now. If you see earlier, we have few compounds, but number of animals were more. There was no restriction on the use of the animals, but now trend has changed. You have to use the animal very judiciously, ethically, with the sound justification. And animal experimentation has to be in the last, after completion of all alternatives being exhausted. When you collect all the information or preclinically, and then you go for the animal experimentation. That is what the trend has changed now. This is what is all about this slides now. Thank you.
Thank you very much, sir, for your knowledgeable talk. Now the session is open for discussion.
Good morning, sir.
Good morning.
This is Mohit. Thank you so much for sharing your thoughts and so valuable information regarding using animals in research. So my simple question, only question is, uh, do you think in near future we would be just bypass preclinical studies for translational research? Can we do that?
We can do it 90%, we can replace it, but in totality animal is to be required because all the informations are computer based. If you say the animals can be totally replaced, it is not possible for the timing.
Okay. So we need preclinical studies at some
Animal
end.
has to be used in the last when you have all information.
Okay. Okay. Thank you.
Thank you very much. Anyone else? Sir, I have one question. If we talk about alternatives like zebrafish, then is it necessary to have that approval by IAEC committee to take?
I see talk about the ethics, right? Even if you are using mosquito, even if you are using microorganisms, if you are using the zebrafish, whatever the experiments you are doing, they just ensure the ethics, nothing else. So there is a no harm putting the experimental proposal through the IAC.
Yeah,
Even if you
right.
publish a paper, you need to have ethical approval. No research journal will publish your paper without ethical approval. No funding
Yes.
agency will give you the financial sanction without ethical approval. Only ethics committee ensure the ethics being followed in the experiment, whether it is a mice, whether it is a fish, whether it is a mosquito, whether it is a microorganisms, anything, any biological organisms that is being used in the experimentation has to be checked or must ensure the ethics. That's why.
Thank you very much,
This is
sir.
very common question. People they ask it. We are using
Yeah,
mosquito. Whether we need to take approval or not. Ideally, it
yeah,
has to taken the approval from the CPCSA.
yeah,
There is no harm in taking ethical approval. They only check the ethics, nothing else.
yeah, definitely, sir. Definitely. Thank you very much, sir. GD, sir, you want to say something?
Good morning, Dr. Saab. Good
Good morning,
morning.
Kasam.
And how are you? So, welcome to the all participants and speakers of today's function, Dr. Anil ji. And most probably, I was 5 minutes late and due to some of the technical clinch. But I feel that number of the participants they are very attentive and listening very carefully. So I request to the organizing committee, Dr. Arti ji, you can also announce one of the award, the person who will give the maximum time or who will attend all the session. Institute will separately announce the best of the like a participant, or we can give the best of the award of like that, because even the international participant they are continuously they are the listening means not that this is the you can say when you are on the platform you can achieve so many things and you can contribute, you can discuss these things, so that number of time we feel that this is the question asked by the Dr. Arti, we can include the zebra fish in the IEC or not. So sometimes we avoid this types of the things, but our one of the students submit the project in the government agency and he send the letter. You can submit the letter through the approval. It means need a some there is the approval. So we need some of the ethics and values. Either we are handling any types of the animals. So sometimes if you can ask the question, or sometime any other person ask the question, or sometime number of the question cleared by the speakers also during the presentation. So I hope that everybody we can achieve something during the presentation. Sometimes we have the content by, sometimes some of the excellent ideas for the future you can develop the research. So thank you very much, Dr. Anilji, and I hope that most of the participants, either they are awardee or participant, they are taking the parts very you can say taking the keen interest. So I hope that in the today we have the three more lecture as well as the presentation of the awardee. So as per the schedule, we will try to complete the whole of the things. and within you can say sometime, and this is the declaration of the, you can say, one of the award of the most sincere participant. Now the question is this one: what is the definition of the most sincere participation? So there is the two definition. One definition is the here, as per the, this is the program is the online mode, so our machine is the recording the complete time. First thing, let us consider you have start your you can click on the link and you can start your video, or sometime you can close the video like that. So first parameter, it will be the time, and second parameter we will ask, let us consider one of the Mr. Mohit Kumarji. In the first day he is the also involved, today he is the also involved. One of the participant I am checking continuously. I think for him, she was full time available even in the inaugural and all vote technical session, and from 10:10 to till time she was available like that. And I hope that this types of the interactive session, it will be the very very fruitful and useful. So Dr. Nirja ji is the also available from the Lucknow. Dr. Nirja, do you want to contribute something?
Ma'am, kindly unmute yourself.
Good morning to everyone. Really, sir, it was a nice session for me. I learned a lot about the animal use of animals. Uh, I'll have a one question in my mind, that you said, sir, ki we use the zebrafish instead of the mice or rat. Is it sufficient that the study, when we changes the animal species, the result is reproducible? When we change the species from mice or rat to the zebrafish or any other, the result will be reproducible?
Ma'am, it depends upon the experiment you are conducting. For example, you are using some experiment. For example, I'll take any example of Alzheimer disease, for example, right? Yeah. When we use any model for Alzheimer, we see what is the pathways, right? Whether these pathways are existing in that model, you are opting. Okay. So same thing, you need to know the uniqueness of the zebrafish.
Okay.
Whether these pathways you are studying is there or not, right? Okay. Certain models have certain limitations, certain unique feature. Okay. So it is very important for the scientists to choose the model based on the receptor pathways available in that model or not. Okay. You cannot blindly say that X model will be applicable in all the disease condition. Okay. It is not possible.
So thank you very much, Dr. Saab, one of the another participant. But first of all, I also, good morning, Dr. Charan ji. He is the away from me, but by heart I have feel that he is in the ISF, and today is the one of the very good day. This is the Guru Parv. So wishing you very very happy Guru Parv to all of you. My good wishes to all. You can grow happy and healthy. So one of the participant is the Avenger. So please. See, yeah. Hands up. So yes, please.
He's an international participant.
Yes.
Ajay, you can.
Yes, my name is Ebenezer Ajayi. Namaste, everyone. I had the privilege to be in India for 2 times, and I think that was when I met Charan and Arti. All right, my question quickly is, sometimes we do improvisation. For example, we want to do bovine. Want to do fetal calf serum or adult bovine serum, or maybe from adult goat. And we are talking about animal handling here. And the people in the abattoir, they don't consider all these things. Because on a few occasions, I have had to improvise, and I had to send my students to go very early in the morning to collect the fresh blood so we could prepare the bovine serum to do tissue culture. Now, if the main animal that gave this blood already underwent some stress, how much control, let me just say, how much control do we have on this improvisation to collect blood from such animals from the abattoir, if you are bringing it under the CP, SCA guidelines? Thank you.
I think it's better, what I could understand from your question is you are saying that your mice or animals are stressed.
No, it was tissue culture, and maybe because for the dearth of funding, we couldn't, at that time when I started, then we could not really raise enough money to buy maybe RPMI or something. So we had to produce our FCS by ourselves by taking animal blood from the abattoir. So I was thinking that if we are using this serum to grow our cells and tissue culture, and the people that will kill the bigger animal, they don't honor anything like handling in a way to reduce stress. We know that some of these things will continue or may continue, probably to modify epigenetically the cells that we will grow at the end of the day. On our side, we will handle the rats very well to get the tissue, but what about the supporting calf serum that we use to grow the animal? When we will take ethical approval, and then we will mention, would they insist that we should go and purchase, or would they want to find any control on this aspect that is not directly under our control? I'm just trying to see, like I saw that there's been an improvement since the last time I was at NIPA in Moali on the guidelines. But does it capture something that is outside of the control of the facility or of the scientist that is conducting such an experiment?
Sorry, I do not have any idea. Sorry.
Yeah, it's about improvisation. Sometimes when we improvise because of some facilities we may not have directly in the lab. That's why I asked the question.
I'm sorry, I do not have any idea regarding this comment.
Thank you very much. Thanks for your time.
So thank you very much, sir. So now,
Arti, can I leave?
Sorry?
Can I leave?
Just wait, sir. Just wait. So thank you very much, sir, for providing us valuable insight on ethics in animal experimentation, especially the 5R concept. And I hope that all the researchers who are working on animal research and those who want to work on animal will find this lecture very fruitful. So we really appreciate your time and effort. And I, on behalf of the organizing committee, want to thank you once again for giving us your time. Thank you very much, sir.
Thank you, Charan. Thank you, Arti.
Thank you very much, sir.
ठीक है आरती, I can leave ना?
Yeah.
Thank you.
Thank you. Thank you very much. Now it is my great pleasure to introduce our second speaker of the day, Dr. Vinod Tiwari from IIT BHU. Dr. Vinod Tiwari is currently working as Assistant Professor of Pharmacology in the Department of Pharmaceutical Engineering and Technology, Indian Institute of Technology, Varanasi. Dr. Tiwari has completed his B Pharm from HNB Garhwal University, Srinagar, Uttarakhand in 2004, followed by M Pharm in Pharmacology from NDMVP College of Pharmacy, Nasik, University of Pune in the year 2007. After completing his M Pharm, Dr. Tiwari has worked in Nicholas Piramal Research Center, Mumbai, where he contributed immensely in the drug development projects related to pain, inflammation, and arthritis. In the year 2008, he joined University Institute of Pharmaceutical Sciences, Punjab University, Chandigarh, for pursuing his doctoral studies. After completing his PhD from Punjab University, Chandigarh in the year 2012, he moved to United States to carry out postdoctoral research at the Johns Hopkins University School of Medicine, Baltimore, Maryland, USA. Before joining IIT BHU, in April 2018, Dr. Tiwari also worked as Assistant Professor of Pharmacology and Toxicology at NIPA, Ahmedabad from 2016 to 2018. Dr. Tiwari is working in the field of pharmacology and neurosciences from past more than 15 years. Dr. Tiwari's research interest is focused on behavioral and molecular neurosciences, dedicated to understanding the various neurodegenerative disorders and the signaling pathway involved so that better and safer drug candidates can be developed by targeting the disrupted neuromachinery. His postdoc research work with Professor Srinivasan N. Raja, Director of Pain Research at the Johns Hopkins University, USA, was focused on dissecting the key neuronal circuitries involved in pain relief and drug abuse so that better analgesic drugs, which are devoid of drug addiction, can be developed. Dr. Tiwari also explored the role of mu delta heterodimers in peripheral opioid analgesia and tolerance, and the finding from this work was recently published in the journal Pain. Dr. Tiwari also contributed immensely in the field of neurosciences with more than 56 research publications, 26 review articles, and 4 book chapters in peer-reviewed high-impact international journals, including Nature Neurosciences, Proceedings of National Academy of Sciences, Journals of Clinical Investigations, Neurons, and many more. His research work is rapidly being cited all over the world with more than 3,700 citations, having H-index of more than 34. He is currently working on different collaborative projects with investigators from Johns Hopkins University, Washington University School of Medicine at St. Louis, USA, and with pharmaceutical industries. His lab is well funded with grants and funded by SERB, ICMR, MHRD, and industrial consultancy projects. Dr. Tiwari has delivered several invited research talks and presented his research findings in more than 50 national and international scientific meetings. He is also serving as editorial and reviewer board member in many leading journals in the field of pain and neurosciences research, along with task force members and reviewers for funding agencies including DBT, SERB, and ICMR. So we welcome you, sir.
Thank you so much, Professor Arti, for the kind introduction. So
thank you very much, sir.
A very good morning to all the
Good morning, sir. Welcome, welcome.
Good morning, Gupta sir. How are you, sir?
Bilkul fine, sir. Fine, sir. Welcome, sir. Yes,
so
continue.
Yes, sir. So first of all, a very good morning to all the participants present over here today, and I would like to congratulate to the organizing team, especially Dr. Charan and Dr. Arti, for taking this wonderful initiative and organizing this neurosciences school at ISF College of Pharmacy, Moga. So without taking further time, I think we are already running behind the time, so I will share my screen with all of you. And I would appreciate, in case you have any queries, any questions, feel free to interrupt me in between and ask the questions. Is the screen visible to everybody?
Yes, sir, it is visible.
Okay, thank you. So in today's, talk, I'm going to talk about the new potential target, which is mu delta opioid heterodimers for the treatment of neuropathic pain. My lab is primarily working in the field of chronic pain, the different signaling pathways, and different, therapeutics for the treatment of different kind of pain disorders. So before I, start my, talk, I would like to pay my tribute to Mahamana Pandit Madan Mohan Maharaji, who is the great visionary, who has developed this beautiful temple of learning where we are sitting today at BHU and IIT BHU. So most of you, attending this talk today might not be knowing that, this is the place, BHU and IIT BHU, where the pharmacy education was started first in our country, not only in our country, but in the whole Southeast Asia. This was the first school of pharmacy. And recently we have completed our 100 years of existence, and Government of India has, launched this, postage stamp, in honor of completion of 100 years of existence of BHU and IIT BHU. So before we talk about the the research component of this talk, let's debate a little bit on pain. What do you think, whether pain is good or bad? Anybody in the audience would like to comment on it?
I think both.
Nobody like to have pain, right? Is it good or bad?
Both.
Both. Good. Thank you. Good. Okay. I know we are going to have diverse opinion on this, so let's go through this quote through, by Swami Sivanand. Oh man, you curse me, blame me, you hate me, and fawn at me. You think I am cruel and heartless. You try to slay me with anesthetics, with chloroform and bromides. You attack me with sedatives and opiates. You phone the doctors. You run to the hospitals. You fly to Vienna and hill stations. You wire to your friends and relatives. You approach the scents of the Himalayas for remedies and herbs. You do Mahamrityunjaya Japa and Havan. You burn incense and pray to kill the teacher who wants you, who comes to help and bless you. I'm not your enemy, I'm your friend. I'm your guide and a silent teacher. I'm pain, the best thing in the world. Why I said so? Look, nobody want to have pain. Whenever we are suffering from, some pain disorder like chronic pain, we go to the doctor for the treatment, right? But just imagine a situation where you are taking a hot shower of water and you cannot feel the pain sensation. You cannot feel how hot is the water. What will happen? You will end up burning your whole skin because you cannot feel the pain because of the temperature, increase in temperature, right? So actually, if you go through the definition of pain, ISP is International Association for the Study of Pain. According to ISP, pain is an unpleasant sensory and emotional experience which is associated with or resembling that associated with actual or potential tissue damage. This definition was recently revised by Professor Srinivasa Raja, who is my postdoc supervisor as well. In 2020 only, they revised this definition. Before that, we were using another definition with like from last 30-40 years. So pain is definitely an unpleasant sensory and emotional experience, but at the same time, it acts as a warning signal. It is a protective mechanism of your body which warns you against some damaging stimuli. If you don't feel how hot is the shower, or how painful is a particular stimuli, you cannot take the proactive step to protect yourself against those damaging stimuli. So pain is also you can say it's a warning signal. It's a protective mechanism of your body. So now the next question is, how do we sense the pain? How do we feel the pain? So nature has given us a wonderful gift. Just like you can taste different kind of food, whether it is salty, sour, sweet. You have the receptors in the tongue, right, which can tell you the taste of different kind of food. In the nose, you have the receptors to smell the different kind of smells, right? In the similar manner, nature has given us different receptors. We also call them pain receptors, or you can also call them nociceptors, because nociception is another term for the pain. So these nociceptors are present all over the peripheral and the central nervous system, and they act as the point of detection, or you can say the place where the pain signal was first detected, and then it transmitted to the other parts in the peripheral and the central nervous system. So if you look at this diagram on the right hand side, let me use the laser pointer. So whenever a person encounters an injury, what happens? The nociceptors or the pain receptors, they get activated, which are present on the peripheral nerve terminals. And these nociceptors, they transmit this pain information from the peripheral nervous system to the DRG neurons. What are DRG neurons? DRG neurons are the dorsal root ganglionic neurons, which are also known as primary sensory neurons, which act as a first part in this sensory system to detect to transmit this pain information, and then they transmit this pain information to the spinal cord, and then spinal cord act as a relay station where this pain information gets modulated and then get further transmitted to the higher pain centers present in the brain, like cortex, amygdala, and other centers in the brain. So this whole red line you see is the ascending pain pathway. And then, the brain, which is our CPU, right, central processing unit, it finally, weighs down the pain intensity and decide whether this pain stimuli, or in other words, you can call the threat, whether this threat or the pain stimuli is credible enough, or not, to take some proactive step against it or not, right? If you're holding some, cup of tea, if it is not very hot, your brain will tell you can hold it and drink the tea, right? But if the cup of water or tea is very hot, you will immediately put it down, right? Because your brain will tell you to do that, right? So this is a descending pathway where our brain judges how credible is the threat, and then based on that, it takes a proactive step to protect us against such damages. Okay, and if you see, here, we do have different kind of receptors to detect the different kind of stimuli. Like if there is a thermal stimuli, hot pain sensation or the cold pain sensation, we have the TRPV1 nociceptor and the TRPM8 nociceptor. When you take the menthol, what kind of feeling you have? Your whole mouth and tongue feels cold, right? So that is because of the activation of TRPM8 receptor present in your mouth and tongue, which is activated by the menthol, right? Or peppermint. So if you go to the Nobel Prize, discovery in the year 2021, that has been awarded to, Professor David Julius, which is from University of California, San Francisco, for the discovery of this, transient receptor potential vanillin 1 nociceptor, which has revolutionized the field of pain research in terms of development of novel therapeutics for the treatment of different kind of pain disorders. So there is another kind of pain nociceptor, which the name is mechanoreceptive nociceptor, which are responsible for the detection of mechanical stimuli, like due to some, like mechanical in nature. So example is MRGD receptor and the TRPA1 receptor. There's another category which is known as the chemoreceptive, nociceptor, which are activated because of exposure to certain chemicals. For example, capsaicin. So that's why TRPA1 nociceptor is also known as the chili receptor, you can say, or the capsaicin receptor, because it activates the TRPA1 receptor. So when you eat a lot of capsaicin, lot of spices, what happens? It leads to the activation of TRPA1 receptor in the mouth and leads to the increase in temperature and the burning sensation, which is because of this TRPA1 nociceptor. Okay, so TRPA1 nociceptor can detect both thermal stimuli and the chemical stimuli, and that is the reason there are certain receptors which can perform more than one function or which can detect more than one stimuli. And that is the reason why we call them polymodal nociceptor. Okay, and the example is TRPA1 and the TRPA1 nociceptor. And when the patients who are suffering from the chronic pain, any kind of pain, whether it is neuropathic in nature or it is inflammatory in nature or migraine or any other kind of pain disorder, so the patient, when it goes to the doctor, it basically complains of two kind of symptoms. One is the mechanical allodynia and second is the thermohypalgesia. Allodynia is basically the term which refers to the increased pain sensation because of the non-noxious stimuli. Non-noxious means non-painful stimuli. Do you think wearing shirt and wearing socks is painful in nature? Not at all, right? But what happens when the doctor goes, when the patient who's suffering from sometimes diabetic neuropathy, chronic diabetic neuropathy, those patients they generally complain of these kind of symptoms. It's very painful for them even to wear the shirt and the socks. So that kind of pain is known as allodynia in nature. And when less noxious stimuli become more noxious in nature, we call it hyperalgesia. And when it is because of heat, we call it thermal. When it is because of some mechanical stimuli, we call it mechanical allodynia. Okay, now in this slide, I'm trying to convince you, pain is not only a symptom, but it is a disease. Unfortunately, most of us, we encounter, different kind of pain, and we do suffer from some diseases which leads to the sometimes chronic pain, but mostly, we think that it is symptom. But actually, if you look at the etiology of the neuropathic pain specifically, you will be surprised to know that, there are different kind of, disorders which are responsible for the nerve injury and neuropathic pain. So, for example, exposure to certain drugs, like, if you talk about toxins, alcohol or the chemotherapeutic drugs, right? Anti-cancer drugs, highly toxic molecules, right? They lead to the nerve damage and lead to the peripheral neuropathy. Chronic alcohol consumption, we all know about the hepatotoxicity, but one of the another, most important complication of chronic alcohol consumption is the alcoholic neuropathy. That was my, PhD research topic also. Then sometimes due to injury, traumatic nerve injury and compression, the patients, encounter the nerve injury and which leads to the persistent feeling of neuropathic pain. Viral infection, for example, the HIV-associated neuropathy. So the patients who suffer from HIV, they have this, issue, another complication, which is HIV-associated neuropathic pain. Vascular-related neurodegenerative, neuropathy is there. Nutritional deficiency. Our, now they are covered with the myelin sheet, and this myelin sheet is made up of, one of the nutritional component, which is vitamin B1. So due to nutritional deficiency, the myelin sheet will become more prone to be attacked by the free radicals, and that may lead to the chronic neuropathic pain. Cancer pain. If you have seen some patients who are suffering from different kind of cancer, specifically bone cancer, right? Their pain is of like worst level. They cry a lot because of the intense pain they are going through. Metabolic disturbances such as diabetes, in case of increased hyperglycemia, the patients have this generation of free radicals, advanced glycogen products, which lead to the nerve damage and the neuropathic pain. Surgical procedures, amputations, in case of the people or the soldiers who are deployed at high altitudes and they are suffering from vascular disorders and like gangrene. And in case of gangrene, surgeons, doctors doesn't have any choice. They just have to amputate the tissues of the patient, soldiers, basically, because they are already rotten because of gangrene. So during such surgical procedures, the nerves also get damaged, which leads to the persistent feeling of neuropathic pain. So if you look at this chart, you will be convinced that neuropathic pain is not only a symptom, but it is a disease which needs the better therapies. So that's why we have mentioned here, it's a common pain state that needs the better therapies. If we talk about the prevalence of neuropathic pain, almost 7% of the general population around us, they're suffering from this disease. And almost 18 to 21% of the patients, who are those who are treated in pain clinics because of the neuropathic pain. If we talk about the impact, it poses a huge social and economic burden on the society and adds to the high healthcare cost and the lost productivity to the nation, because these people cannot contribute to the economic growth of the society because of their inability to work. If we talk about the treatment, unfortunately, the treatment is really difficult. We do have some drugs in the clinic, like opioids and some other drugs, but unfortunately, most of these drugs, they provide inadequate pain relief. Only 20 to 25% of the patients are responding to these therapeutics. That means almost 60 to 70% patients are still remain untreated in spite of having so many drugs in the clinic. If we talk about the opioid-like drug, specifically morphine, which we know that is one of the most potent analgesic available with us. If you go to the Goodman-Gilman, you will find out that it is mentioned in the book as well. Morphine is God's own medicine, very hard to beat, and it's remained one of the most widely used drug. So if we have such a wonderful drug in the clinic, why we need to go for the search for better analgesic? The reason is, the issue is the undesirable side effects and the tolerance which is associated with the chronic use of opioid-like drug. If you go to the literature, you will come to know that morphine-like drugs, in spite of having the potent, analgesic efficacy, they produce a lot of side effects and toxicities because of activation of mu-opioid receptors present in the central nervous system and in the peripheral nervous system. It leads to sedation, dizziness, respiratory depression, addiction, and constipation. That is the reason why opioid-like drugs, they have been degraded, from first line of therapy to second and third line of therapy. So this is some of the literature for your reference, where it has been mentioned, and these side effects and toxicities are not the minor issues. They are highly challenging issues for the clinicians, and they are struggling with it a lot. So you see, these drugs lead to sedation, respiratory depression, and people have given it a specific term, which is known as opioid crisis, because the patients, sometimes they die as well because of the overdose of opioid-like drugs. So that is the reason why, people are, scientists are still, working in this direction to develop the better molecules. Now, the question which we asked, in our lab, can we develop better therapies for neuropathic pain treatment, which will be devoid of all these side effects and toxicities? So in the light of these challenges, our efforts was focused on, the opioid receptor signaling and can we develop some, new molecules. So we came across some literature where people were working on opioid receptor heterodimers. We know what are heterodimers, right? When two different kind of receptors they merge together, they work with each other, we call it heterodimer basically. When the similar receptor they interact with each other, we call it homodimerism, right? So in this case, if you go to the literature, you will come to know that there are a lot of studies on mu and delta opioid receptors and their interaction. Some people have reported their role in the opioid tolerance and some other issues. So but what we found actually that these heterodimers, because they are also the part of the G protein coupled receptor family, they share a very unique pharmacology, which is somewhat different from the conventional, like mu receptor and the opioid receptor or the delta opioid receptors. So we thought that, let's explore this, heterodimer, for the analgesic efficacy, whether they can act as a, target for the potential pain relief or not. But unfortunately, in the past few decades, that was not possible because of the lack of specific tools to activate this heterodimer and to, probe down this heterodimer basically. But I would say, in the last few years, one of the lab, in New York, which is our collaborator as well, Professor Lakshmi Devi. So I would like to show their lab, if it is here, yeah. So they have developed one antibody against the mu delta heterodimer. Professor Lakshmi, their lab has developed. So which has really, helped to probe down this field, in a much detailed manner. So where we were able to, actually, investigate the expression of mu delta heterodimers. We'll come to this data later on. So yeah, so the advantage of targeting the mu delta heterodimer is that we thought that we can provide a more potent and long-lasting inhibition of neuropathic pain. The second advantage which we felt is there will be less or the no off-target adverse effects, because we thought of activating only the peripheral, mu opioid receptor or the peripheral, heterodimers. So with this objective in our mind, we have used one drug, dalda, which is a peptide drug. It's not the ghee dalda which we use in our home. It's a peptide drug basically, which activate the mu opioid receptor only. So yeah, it's a peripheral mu opioid receptor agonist. The another drug which we used in this study is the CYM51010 molecule, which is a activator of the mu delta heterodimer. So this molecule was also recently developed. So these two discoveries, the antibody for the mu delta heterodimer and the development of this 1010 molecule, has expedited the field of this heterodimer discovery recently. So yeah. And then we further went on to probe down the cellular and molecular mechanisms which are associated with the this activation of mu delta heterodimerism. So in this study, we have used the male SD rats and the male wild type mice and the mu and the delta opioid receptor knockout mice. We have used the SNL model, which is spinal nerve ligation model. So in rat, we have to ligate the L5 spinal nerves. If you are a little bit aware with the anatomy of the these spinal nerves and the sciatic nerve, from the lumbar region, different spinal nerves they come out like L1, L2. So sciatic nerve is a combination of L3, L4, L5, L6 nerve, and they form the sciatic nerve. And then the sciatic nerve, further trifurcate into three different nerves, which is not shown here: peroneal nerve, tibial nerve, and the sural nerves. So these three nerves, they supply our hand limbs, and in case of, like legs in case of humans, right? In case of animals, hand limbs. So, but in case of mice, the anatomy is slightly different. So to induce the model of long-lasting neuropathic pain, we ligate L4 nerve instead of L5. Okay. And then we did another surgery in the animals, in the rats specifically, is the intrathecal catheter implantation surgery, in which we implant PE10 tubing in the lumbar region of the spinal cord of the rats to deliver the drug specifically into the spinal cord of the animal. Then dalda, I already mentioned, it's the peripherally acting mu opioid receptor agonist, and 1010 molecule is the activator of the mu delta heterodimer. And then morphine also we used in our experiments to compare our data, and also to develop the model of tolerance to see what is happening with these animals under the morphine tolerance. This is the first set of data which I would like to discuss with all of you. So in this study, we were interested, first of all, we wanted to understand, what is happening under the nerve injury condition with these mu delta heterodimers, whether their expression will go down or will go up. So interestingly, we L5-DRG neurons are the injured neurons and L4 are the uninjured. So DRG, dorsal root ganglionic neurons, basically they are sitting just next to each other. So interestingly, when we did the immunohistochemistry in the DRG of the animals, with and without nerve injury. So this is the data from the non-injured neuron. This is from the injured DRG neurons. So in the red color you see is the mu delta dimer receptors are present, and in the green color is for the mu opioid receptor basically. And we merge these images, we can find out how many mu opioid receptors are co-localized with the mu delta heterodimers. So after quantifying this data, what he found actually, if you see in case of control animals, control means the uninjured animals, the naive animals, we did not find any difference in the heterodimer levels. Okay, in case of control, animals, basically both in L5 and in the L4 DRGs. Okay, and then but after the nerve injury, what we found that there's a significant increase in the dimer positive neurons in the basically, this is the L4 DRG, uninjured neurons. But there was a decrease in the mu delta heterodimers in the injured DRG neurons. Then we went on to perform to confirm these findings by using the ELISA study, where we incorporated one more group, which is the DALDA treated animals, because it has been reported that, because in one of our study we found that when we give the DALDA, for a repeated period of time, it created the tolerance. And some people have suggested, in literature that the tolerance, the opioid tolerance is associated with the increase in heterodimerization. So that thing also we wanted to confirm in this study. So if you see in this data, in ELISA also as well, we found that there was a significant increase in the mu delta heterodimers in the L4 DRG neurons, which is uninjured, while decrease in the L5 DRG. Okay. And but under the tolerance like condition in the animals which are subjected to the repeated DALDA administration, but under nerve injury condition, the increase was significantly more, much increase, which is like way more than the nerve injury alone. So both in case of L4 and the L5 DRGs. That was the finding which kind of gave us a hint that there is something which is going on under the tolerance condition, the opioid tolerance condition. Okay, then first of all we decided to test the activity of this mu delta heterodimer agonist in the nerve injured animals itself, that is after the SLS surgery. So if you look at this data, this is the mechanical pain behavior which has been tested by using the worm-free hair test, and this is the thermal pain behavior where we measure the power-to latency of the animal by using the Hargreaves test. So we used 2 doses of the 1010 molecule, 0.2 mg per kg and 2 mg per kg. Interestingly, at a dose of 0.2 mg per kg, we did not observe any significant increase in povital threshold. If you look at this data, this is the povital threshold on the y-axis, and on the x-axis you can see the different time points. Like pre-SNL means the pre-injury. So before injury, the povital threshold was around 20 to 25 gram, which indicates a higher povital threshold. That means animals are not withdrawing their paw at a higher force. But after the nerve injury, that is day 7, after the SNL surgery, after the nerve injury, the povital threshold of animals, it dropped down to around 5 gram, which indicates that animals are experiencing intense pain when they are subjected to the worm-free hair filaments. And the same happened in case of Hargreaves apparatus when they are exposed to the this thermal stimuli on the subplantar region. The povital latency was significantly dropped from 15 seconds to almost 6 to 7 seconds. So this indicates that the animals after injury are exhibiting significant hypersensitivity. And when these animals were treated with different doses of 1010 molecule, we found that only 2 mg per kg of the dose, which is 10 times more than 0.2 mg per kg of the dose, was able to attenuate or decrease the pain behavior in these animals, in nerve injured animals. And then we after that we ran the whole dose response curve for 1010 molecule at different doses, and we found that at 1, 1.5 and 4 mg per kg the drug was able to restore the pain behavior. Then we also calculated the EC50, which came around 1 mg per kg, And then we also performed the rotor route test just to confirm whether this molecule is suffering from any CNS toxicity or not, just like morphine like drugs. So at this dose at least 2 mg per kg, the highest dose, the 1010 molecule, does not lead to any decrease in in the fall time of the animals, which indicates it does not lead to the motor coordination. Okay. So after this, because we are more interested to understand, what is happening under the opioid tolerance condition. So we have developed the animal model of morphine tolerance. If you see
I'm
this,
sorry, Dr. Vinod, to interrupt
please.
you in between. One of the candidates has raised the hand.
Yeah, sure, please.
Yes.
So I have some question regarding that heterodimers, the opioid receptor heterodimers. So are these receptors only available on neurons or any other kind of cells?
So yeah, these heterodimers, they are present all over the, like in the peripheral nervous system, in the central nervous system, and even in some of the brain regions, basically. But what happens
in spinal cord?
Yeah, they are present in the spinal cord as well. So they are present everywhere. But the thing is that under certain pathological condition like nerve injury or some inflammatory condition, or under some treatment condition like after opioid treatment, their levels, they do get changed. And that is something which we wanted to investigate in this study, like what is happening after nerve injury and what is happening under the tolerance-like condition.
So my question is like, how are you so sure that the slide you are showing that in heterodimer and that new opioid receptors and heteromeric, they are merged equally and they are actually on the neurons, not in any other kind of cells?
So we have used the primary antibody against the DRG neurons, okay, and we also did some of the other studies. The data I'm not showing here basically, which confirm that, like NEUN, for example, right, which confirms that these receptors which we are looking at are present on the neuron. So look, we are not bothered whether these receptors are right now present on the neuron or the non-neuronal cells, right? Our main concern is that whether the expression of these receptors is get elevated after the nerve injury or after the opioid tolerance. This is what we wanted to understand.
Okay. Thank you.
Okay, so in this study, if you look at the data, first we have created the animal model of morphine tolerance. If you see, the pivotal threshold of the animals before injury was around 20 gram, right? And then after the nerve injury on the day one, which is like, you can say after 7 to 14 days after the nerve injury, the pivotal threshold was significantly dropped to around 2 to 3 gram actually, which clearly indicates that the animals are, having intense pain. And when we treated these animals with morphine, the pivotal threshold goes to again around 20 gram. We all know that morphine is one of the most potential analgesic, right? And these for this, indigenous tolerance, we injected morphine 2 times a day continuously for the 7 days. So if you look at this data, you will, one second, let me use the laser. If you look at this data, you will, you can easily see that the potency or the efficacy of the morphine day after day is continuously, decreasing. So which indicate that these animals, actually, are developing tolerance to the morphine. And when we tested their, pivotal threshold on day 9, the morphine was unable to produce any analgesic effect, which indicates that the morphine are completely tolerant to morphine right now after 7 days, twice a day treatment with morphine. So then, why we created this model? Because we wanted to test the effect of 1010 molecule, which is a heterodimer, agonist. And in one of our previous study, we found that, especially under tolerance conditions, these heterodimer levels, they got increased. Okay. So if you see in the panel B, this is the pre-SNL baseline. And we again use the 1010 molecule in the same doses, 0.2 mg per kg and 2 mg per kg, right? And what you see in the x-axis is the pre-tolerance data and the post-tolerance data. That means this is the effect of the 1010 molecule before morphine tolerance in these animals. And this is the effect of 1010 molecule in the same animals after the induction of tolerance. So if you see at this data, look at this data, you will see that 0.2 mg per kg of the dose again did not produce any significant effect. But
sorry for the interruption, sir. Sorry for the interruption. We have our faculty, Dr. Shamsher. He want to ask.
Yeah, please, Dr. Samsher, please go ahead.
Yes, sir. I am having one question that you are
Please.
using a morphine instead of apomorphine. So why you are preferring morphine instead of
No,
apomorphine?
morphine is an analgesic drug. Apomorphine is a drug which generally used for.
Basically, morphine do not correlated with the ipsilateral and contralateral. If you see,
Look. I think you are talking about some other, disease. If you go to the
not in other disease. If you give any morphine to any animal, there will be some alter behavior that will be related with the peripheral movements. So
Okay.
because if we do like suppose if you want to do this plantar aesthesiometer, in that case might be the morphine administration can alter the behavior of that animal.
Look, Dr. Samsher, in the pain research, actually, the clinician, the doctors, actually, the patients who are suffering from chronic neuropathic pain disorder, morphine is one of the most widely used drug, okay? Because it works through the opioid receptor and it suppresses the pain, basically, right?
Basically, for the chronic pain, yes.
Sorry?
For the chronic pain, it is preferred.
Yeah, for the chronic pain, morphine. So morphine is the most widely used drug. And
but
that is
my question is, suppose if I will say that morphine induce ipsilateral and contralateral behavior in animal, how you can confirm
what kind of ipsilateral and contralateral behavior?
That mean ipsilateral mean it will be the movement of the animal either toward the left side or right side. Suppose that is commonly appeared in case of CNS disorder.
So that is not associated with the morphine, actually, I think you're talking about apomorphine right now.
That is with the apomorphine. So is there any correlation that whether the animal shows such type of movement?
No. In morphine, we did not observe those kind of activity. So morphine does not show those kind of. You're talking about the stereotype behavior, I know.
Because it is converted into that derivative also after the metabolism. Yes.
Yeah, I agree.
That's why, because either you will give morphine, ultimately in the body it will be appeared as a metabolite form.
No. But morphine, morphine treatment, we are using morphine from past 15 years. We did not observe this stereotype behavior with the morphine. It is specifically linked with the apomorphine only.
Okay. Thank you.
Okay, so let's come to the thermal hyperalgesia data in which you measure the povidone latency of the animals. To our surprise, what we found that before the induction of tolerance in these animals, the 0.2 mg per kg of the dose, which is gray circle, actually does not induce, any, analgesic activity. But after the induction of tolerance in these animals, what happened, even the 0.2 mg per kg of the dose was started showing the analgesic activity. So that was a kind of, finding which, surprised us a little bit at that time. But we had some idea from our previous findings from the immunohistochemistry study where we found that under tolerance condition, the levels of mu delta heterodimer, they get increased. Then we also performed another test for the assessment of spontaneous ongoing pain in animals. So recently, the scientists, how much time do I have, Dr. Arti?
You can continue, sir.
Okay, thank you.
Yeah, you can continue.
Okay, so recently, there was a lot of debate on in the pain research field, like regarding the behavior assessment of pain, what scientists are doing currently. So most of the pain behavior is associated with the evoked stimuli, where we provide certain kind of stimuli, for example, warm-free filaments, or we provide some kind of thermal stimuli to the animal, and then we give the drugs and we see whether the drugs are producing, decrease in or increase in povidone threshold or latencies. So scientists were debating a lot, because in case of patients, sometime when even they are taking rest, who are suffering from chronic pain, we are not providing any kind of stimuli, but they still complain of pain, right? So a lot of people, they came up with different kind of behavior assessment of pain, but this is one of the most validated one recently, known as condition place preference. So initially this test was used for the drug addiction studies, where people used to see which chamber will animal prefer after treatment or pairing with certain kind of drug. So but Frank Poreka from University of Arizona, he's a professor there and he's our collaborator as well. So Professor Frank Poreka, he used this apparatus and modified it for the assessment of spontaneous ongoing pain in rats. If you see this picture on the right hand panel, on the topmost panel, basically this apparatus consists of two different chambers, including one corridor, And the two different chambers, they are having a different texture that for the tactile stimuli, and different textures on the walls as well. So animals have a different tactile cue on the bottom and different visual cues on the wall, so that animal can distinguish between both the chambers. So it is a learned behavior in many vertebrates, including humans, where we can use this to figure out when the animals or the subject prefers one place more than others, because the preferred location has been paired previously with the rewarding events such as pain relief. So what we do in this test, basically, before the post conditioning test, where this corridor is always open and animal can choose either of the chamber. So in the preconditioning, animal can choose either of the chamber. So generally animals don't have any preferences because they have not been paired. But during the conditioning, what we do, in one of the chamber, we provide the drug treatment. And if the drug is having analgesic activity, the pain relieving effect, and we lock the animals actually in this chamber for 30 to 45 minutes. So if the drug is having an analgesic, activity, the animal can remember it actually, and can correlate it with the pain relieving effect, the environment of the chamber. And after this, we again pair the animals, in the morning, first half, with the vehicle, which is not supposed to have any activity, in the another chamber. And in the post conditioning, we remove the barrier from the corridor and we allow the animal to explore any of the chamber. So that is done on the another day. And then we calculate the time spent by the animal in different chamber, like chamber A versus chamber B, or you can say the chamber which is vehicle paired versus the drug paired. So if the drug is having an analgesic activity, the animal will spend more time in the drug paired chambers. And this is what exactly we found here, So we use the 10 molecule at a dose of 2 mg per kg. And we found that, the difference, here we have presented difference score, in which we subtracted the time spent in the vehicle paired chamber from the drug paired chamber. So the difference score was more, which means the time spent in the drug paired chamber was more in these animals, nerve injured animals. Interestingly, when the animals were, these are nerve injured animals. Okay. Now someone can debate, like this can happen in the drug addiction condition as well, right? When the animals are, when the molecule is having the drug addictive potential, then also animals are going to show this kind of preference behavior, right? So to answer that question, we have used the uninjured animals as well, in which we have not done any surgery, we have not done any nerve injury, right? And in these animals, we do not observe any place preference behavior, which clearly indicate that the animals are exhibiting place preference behavior only those animals which are nerve injured. So this rewarding effects are independent of, or you can say are dependent on the nerve injury condition. Okay. Then in the next set of study, we have, tested, the effect at a lower dose, which is lower dose.
Sorry for interruption, sir. I think one of the participants raised hand.
Please, sure.
Sir, can we say that by this experiment that this drug, the CYM010, doesn't have any rewarding effects?
No. If you look at the data, Mohit, just look at the data. So, yeah. So here, if you see, when we use this molecule at a dose of 2 mg per kg, we found that, the difference score was high, which indicate that the drug, the animals are showing, more preference for the CYM, treated, in the paired chamber, basically, right? So, but when we tested the same dose of this molecule in the uninjured animal or the naive animal, there was no rewarding effect. You can see the difference score was almost non-significant.
That means drug is not binding to any region in the brain, but specifically to the spinal cord and DG neurons and showing their effect.
It's not like that, Mohit. So yeah. So in one of our study, what we found actually, when the drug is only peripherally acting, that means the drug doesn't go to the brain. It cannot cross the blood-brain barrier, right? So it only activates the peripheral mu opioid receptor. Under that condition, also drug was able to produce the place preference. So the place preference behavior is not because of the activation of the receptors present in the brain, but it's just because of the feeling of the pain relief. So whenever we feel rewarding, right, when we get some promotion, some prize, right, is because of the activation of the reward circuitry in the brain. Like ventral tegmental area, nucleus accumbens, these are the reward regions in the brain, right?
Exactly.
So there is a release of the dopamine in the brain, right? So that release in the dopamine can be because of activation of receptor directly in the brain, or because of some of the like activation in the peripheral nervous system as well. And that will lead to the pain relief, and that pain relief is going to activate the reward circuitry in this case.
But
So
in
here,
your case, yeah, in case of your drug, it's not happening in the brain.
yeah, in this case, why we did this experiment, you might be getting confused with the new data, right? So in this case, why we performed this study, because once a molecule produce the place preference behavior, anyone can argue that this place preference may be because of the addiction potential, not because of the, because animal may show the preference because of the pain relief induced reward, or because of the activation of the addictive, because of the addiction potential as well, right?
That's like morphine.
Yeah, like morphine.
Yes.
Exactly. We did that study as well, but the data is not here today. Okay, so if you see at this data, so in case of naive animals, we did not observe any place preference. So which clearly indicate that this molecule only produce the place preference in the nerve injured animals, SNL animals. So the place preference is dependent on the nerve injury, not on the, okay. So it's not because of the if it is addiction, then it should have produced this place preference in the naive animals as well.
So did you also check the withdrawal effects as well with this drug?
No, we haven't tested
okay,
that.
Okay. That would be interesting too. Sorry.
Yeah.
Thank you so much.
Sir, we have one more participant, Shilpa. Yes, Shilpa, you can ask.
Hello, good morning, everyone. Good morning, sir.
Switch on your camera. Let's switch on your camera.
Okay.
Yeah.
Okay, good morning, sir.
Good morning.
Sir, actually, as in this experiment, I can see somewhere memory is involved. Have you performed any cognitive function test or memory test here?
No, it was not required basically, because that was not the objective of a study. Yes, but you are right.
But
in case
like for place preference or like for reward functioning, there is involvement of memory.
Of course, there is involvement of memory, but our objective was just to see, whether the animals can show the place preference behavior or not.
Okay, but if your animals are cognitively impaired or impairment of memory is there?
That was not the objective of the study. And we have not induced any cognitive defects in these animals. We have just induced the nerve injury for induction of neuropathic pain.
Okay.
Yeah. So that was not the part of the study. That's why we have not done it. But if somebody is interested to look on those aspects, definitely they can do it.
Okay. Thank you so much, sir.
I would be interested in reward neurocircuitry.
And the PPT is...
Yeah.
Yes, sir. You can continue.
Yeah. Okay.
Yeah.
Okay. So that was all about the the spontaneous ongoing pain data. Then we wanted to study the mechanism behind this phenomena. What is happening? Why this 10-10 molecule is able to produce the pain relief at a much lower dose, which it was not exhibiting. Actually, if you have seen the data, under nerve injury condition, 0.2 mg per kg of 10-10 molecule was not showing any activity initially, right? But under the tolerance condition, when the animals are tolerant to morphine, both in case of condition place preference and in case of mechanical pain behavior, the 0.2 mg per kg dose started producing the analgesic activity. So we were wondering what is happening, at the cellular and the molecular level. So to address this question, we have done the this study where we have used the HEC293 cells, and we checked the mu-opioid receptor with the green color, and we tagged the delta opioid receptor with the MIC, which is red in color. If you see this data in the control picture here on the left hand side, let me use the pointer. Here, most of the green color and the red color you will find in the periphery of the cells, HEC cells basically, right? So which indicate that most of the mu-opioid receptors and the delta opioid receptor, they are present, mostly on the periphery and somewhat in the, inside the cell as well, right? But when these cells were incubated with the DAMGO is a mu-opioid receptor agonist. So we incubated these cells with 5 micromole of DAMGO, and after 45 minutes, we found that, these mu-opioid receptors, they start migrating inside the cell. And they're not only migrating, alone, they also, brought the delta opioid receptor, which are, tagged red in color, to inside the cells. So which clearly indicate that under the, mu-opioid receptor activation condition, when we incubated, these cells with the mu-opioid receptor agonist, it will lead to the internalization of the or the co-internalization of the delta opioid receptor as well. And the same in case of DALDA, which is another mu opioid receptor agonist. And same data we obtained with the CMY5 1010 molecule. That after, so this data clearly indicate that after activation of mu opioid receptor, it leads to the co-internalization of the delta opioid receptor, which were present initially on the surface of the cell. Okay, and that indicate that this mu opioid receptor and delta opioid receptor, they have some kind of functional interaction with each other. You are activating one receptor, and then that is leading to the movement of another receptor with it. So that clearly indicate that these receptors, they do have some functional interaction with each other. Okay, now in the next set of study, we wanted to understand what is happening, because all the neurons, all the cells in our body, they have their own language of communication, just like we are talking to each other in terms of like English or Hindi or any other language, right? The cells, they also communicate with each other in terms of electrical energy, in terms of action potential, right? That you all know very well, right? So there are three different fibers which are present in our peripheral nerve terminals: A beta fiber, A delta fiber, and the C fiber. So these are the different fibers which are responsible for carrying the pain information from the peripheral nervous system to the central nervous system. So and there is one phenomenon, wind up. Wind up means during the basically chronic pain condition, there is hyperexcitability of the excitatory neurotransmitters such as glutamate receptor is there. So they lead to the train of events where there is generation of lot of action potential, which lead to the increased hyperexcitability and wind up, which leads to the feeling of persistent pain in the humans and in the animals as well, right? So we wanted to measure the activity of all these fibers, and we did the electrophysiological study for this, in vivo electrophysiology. Okay, so where we anesthetize the animal and we stimulate the animals, and then we are recording from the lumbar region of the spinal cord of the animals, and the animals are alive but they are under anesthesia. So we did measure the A alpha A beta component and the A delta component as well. So this is before the treatment with 1010 molecule, which is agonist of mu delta heterodimer, and this is after the treatment with 1010 molecule. We could not find any effect. If you see this line, the hollow circle and the black circle, they are running together, right? Both in case of A alpha beta component and both in case of A delta component. So the action, there is no change in the action potential after the treatment with 1010 molecule. Okay, but when we measure the C component, we found that there was a significant decrease in the C component of the action potential after the treatment with 1010 molecule. And when we quantify this complete data, we can easily see that there's a significant drop in the total action potential of the C component actually. Then we also measure the wind up function, where again we found that the total response, and all these things are done in the nerve injured animals. Total response was significantly decreased after the treatment with the 1010 molecule, but that is specifically associated with the C component only. Then we went on to to further confirm these findings in the knockout animals, because this molecule is a agonist of the mu delta heterodimer. So we thought let's just confirm whether this molecule can still retain its analgesic activity if we delete one of these receptors from the animal. So to answer this question, we have used the global mu opioid receptor knockout animals. Now I think you must be having an idea about the tissue specific knockout and the global knockout. Global knockout means that particular receptor has been deleted from all over the body of the animals, right? But tissue specific knockout means we have deleted that particular gene or particular receptor in a specific tissue of the body, like DRG, spinal cord, or some region of the brain. But these are the global knockout animals. Okay, so if you look at the panel A, this is the worm-free hair test, where so this is done in the mice. In mice, we measure the povital frequency. Okay, where we just quantify the data in terms of how many times the animal withdraw the paw after the activation with the worm-free filaments. Okay, so if you look at this data, the open circle and the black circle, right? So this is the pre-injury, this is pre-drug. Then after treatment with the drug, the animals which are wild type, which is the white circle, the open circle, right? the animal's povital frequency is significantly decreased, which suggests that the drug is having the analgesic activity because it is decreasing the povital frequency. But unfortunately, if you see, look at the data of the knockout animals, which is black circle, the animals, the povital frequency is still same before and after the treatment, even at 1 hour and 2 hours after the drug treatment, which indicate that these animals which are knockout with mu-opioid receptor are not responding to the mu delta heterodimer agonist, which is 10-10 molecule. And this happened in case of, like this is ipsilateral, which is the injured paw, and this is a contralateral, which is the uninjured paw. So when we measured this behavior at a high force, which is 0.4 gram, we found the same result. And then we measured the povital latency by using Hargreaves separators. Again, we found that after the deletion of this mu-opioid receptor, the analgesic effects of 10-10 molecule was lost, which clearly indicate that 10-10 CYM molecule require.
हेलो
हेलो आई थिंक सर टेक्निकल इशू है इस वजह से वी कैन वेट फॉर सम टाइम So meanwhile, those who are having the presentation session, they can join via the link which is shared through email and WhatsApp. So that presentation part is going on through different link. You can join that part, but this is only for those bodies. Yeah. Yes, Vinod.
Sorry for the interruption. There was some network glitch on my end. So fortunately, it did not happen during the talk. Sorry about that.
It's
no
okay,
problem, sir.
No problem.
So please feel free to ask any questions.
Sir, is presentation done from your side?
Yes, it was done. So
okay, definitely.
I'm really sorry. I think
thank you very much, sir.
There was some network glitch, I think, from my side.
Okay, it's okay, sir. So thank you very much, sir, for your informative talk. Now the session is open for discussion. Yeah, now the session is open for discussion. If any participant want to ask or discuss anything. Okay, we have Charan Singh with us. Dr. Charan Singh, you want to share?
Thank you, Dr. Arti. So, sir, good afternoon, directors and Dr. Vinod Tiwari, because this is from my side, like I directed you earlier. So I'm happy to have to know about the research, Dr. Tiwari, from your side. As you have said, this is outcomes of 15 years, and that you have, reached up to here regarding the same thing. So this is a, good thing, I'd like to say. So this is up to the participants if they want to ask something from their side.
Thank you very much, Dr. Charan. Judy sir, Dr. G.D.Gupta. Sir, you want to share some views?
I hope if any participant they want to interact, I know that this is not my field of the research, but I feel that the most of the things very gentle technical manner. Dr. Vinod Tiwari ji is the justified, and during this presentation, I was also on a talk with the one of the hospital, and we are talking about some of the different issue, and this is related to the bath, pitta, and kapha. And I feel that even we are the working on the so many science and so many technology, what is the need of the society, this is the even is the somewhere is the space. So if any person is the doing the work in the neuroscience or any types of the drug delivery system, or they are the handling of the animal, so I feel that they are also required some of the new development in the model, as well as the what is the actual problem in the society nowadays, when the climatic condition is the change, and so many problems, allergic condition maybe take place, and person is dependent on the you can say citrizine, levocitrizine, like that. So even that I know that in those days market of the citrizine, it will be the very huge, and no any other alternate even that is the available or in the market. So I hope that the young scientists, they are the work on the so many derivatives we are the synthesizing in our lab. So we need to the test of the more and more. Second part is this one. This is from my side request that when any speaker they have the delivering the lecture, so they have some of the pace. When any audience asks the question, so pace is the breakout, and then it is very difficult to the come on the same of the pace or same of the like that. So I request to the all, you can write down your question, and you can also write down the if you have some question in the specific slide, you can also write down, so organizer also will take care. And at the end, any person hands up, we can write down, and end of the lecture, one by one we can take the inquiry, and it will be the give the huge of the interactive session, so that we can find out the more informative things like that. Because in the intermediate sometime what I feel that sometimes it will be the create, because what types of the flow we are the going on, but when this flow is the break, then sometime number of the examples, number of the things, it will be the missing out. So because in the next time speaker will be the change, but we all are on the same platform, so we can the note down. And before to the start the presentation, we can also request to the speakers, those who will the present next session, they have also mentioned the slide number. Let us consider I have some on inquiries on the slide number 5 or like that, so we can ask easily. So I hope that this will be the more effective. And Dr. Vinod Tiwari lectures, number of the person they are involved, and they are cleared their queries. So this platform is the open to all. You can ask the question, you can discuss. Sometimes you have some doubt, sometimes you feel that if your research is the, you are the more interested in the research, and your field is the same, and you have did the lot of work, so you can also contribute. And one question is the asked in the previous lecture. This is the reproducibility with I think this is the asked question by the Dr. Nirja. So the reproducibility in the zebra face or like the mic. So what I feel that if you are the performing in the work in the zebra face, and if you can the same perform the work in the rat or mice, so there is the no any you can say relation as a reproducibility. Reproducibility in the same types of the animals, and results will be the same. So model of the zebra face, and limitation of the zebra face, and some of the activity of the zebra fish, they are slightly the limitation, but open field is the rat and mice. So we can perform when we are talking about the bioequivalency, then this is the same thing. We need to the same doses form, but manufacturer will be the difference, and we can find out the bioequivalency test like that. So excellent, and I hope that everybody is the satisfied with this the lecture, and you can also take the screenshot, and you can also share the screenshot in your group, so that person they can be those who are not involved, they can also interact and ask the question during the thing. So I hope that this is the pure basically in the pharmacology based or the neuroscience, neuro disorder like that. So those are doing in this field, they are more and more, they will be the highly beneficial like that. So thank you very much, and thanks to the organizing committee, especially Dr. Arti ji. She is taking a lot of pain and their team to organize the such types of the lecture in minute to minutes. And I hope that the second session is the session, I think the third one, presentation of the awardee. So during the presentation of awardee, I request to the participants, you can come with the third session with the full of energy and enthusiasm. And don't worry that if we have the time, we have also give the opportunity to you what you can involve in the interactive session during the presentation. So thank you. Thanks to all.
Thank you
So,
very much. Yeah.
Dr.
Yes,
Vinod.
doctor.
Yeah, actually, one of the participants have one question. What is the relation between diabetes with respect to neuropathy brain?
Sonima has asked this question. I have seen that. So, Sonima, you know what happens? Can you hear me, Sonima? If you are here. Okay. So,
Yes, sir.
So what happened actually during diabetes condition? You know, there's hyperglycemia, right? There are increased blood glucose levels, right? And when this glucose is not able to, get into the cell, what happens? It chooses the alternate pathway, right? And there's a formation of, advanced glycogen end products, right? And there's a lot of free radical generation of, nitrosative stress, at the same time, generation of pro-inflammatory cytokines. So all these things, they lead to the peripheral nerve damage, and that lead to the central sensitization and neuropathic pain, diabetic neuropathy. Okay.
Thank you, sir.
Thank you very much, sir. The session is full of healthy discussion. So thank you very much, Dr. Vinod Tiwari, for providing informative talk on painful research, on pain research, And I think it is very much helpful for those who want to work in the pain research. So thank you very much for your time and effort.
Thank you so much, Dr. Arti, for really taking this initiative to conduct all this program, and thanks to Dr. Charan, for bringing this workshop to ISF Moga. Thank you so much.
Dr. Arti ji, you have especially mentioned pain research or pain. You have used the word, so research is always painful, but their output always very, very fruitful. So if you take the more and more pain in your research, then you will be the happy and flourish in your throughout the life.
Definitely, sir. Definitely. You're true.
Thank you. So can I leave now?
Yes, sir.
Thank you, sir.
Thank you so much, sir.
Thank you.
Now that's all from the technical session of the half-day technical session. Lecture we have completed, and from 2 p.m. onwards, we'll start our third lecture of the day. So you can from now you can leave the meeting and you can join at 2 p.m. for third lecture.